Alrighty, so we are going to start with our phylum Porifera notes, just a little bit about sponges as a whole. So when we think about Porifera, Porifera means pore bearer, and it's exactly what sponges are. So they have tons and tons of little pores all over them, and that's what helps them to kind of be a filter feeder. So when we start talking about a marine biology and we start talking about these invertebrates that we're going to study, we're going to look at them as they are organized in terms of very simple organisms all the way up to much more complex organisms. So we're going to start with the organization level of being cellular. So sponges are organized at the cellular level, meaning that they just have a bunch of little specialized cells inside of them that make up the organism as a whole. So they don't have cells that build tissues and tissues build organs and organs build organ systems. They basically are just an actual organism, an actual animal that is organized with just different types of cells. So in theory, if we put sponges into a cellular blender, we didn't damage any of the cells, we could split them up into all these different little types of cells, and then they would recongregate back into this really cool organism itself. So they do have asymmetrical symmetry. So when we look at a sponge, we can look at a couple different species of sponges and see that our asymmetry is very true of all of our different types of sponges. We cannot divide these out. This is actually just a portion of one. We cannot divide them out into a perfectly symmetrical organism. So they have that asymmetrical symmetry. Now, adult sponges are sessile. So sessile is a term that you do need to know, and it just means to be attached to a surface. So when we talked about some of the characteristics of animals, we talked about how animals at some point in their lives are mobile. So they can move some time during their existence between birth and death, but it may not be their entire existence that they're moving around. So sponges are actually sessile as adults, and then their planktonic larvae, so when they're baby sponges, that's when they kind of float around in the water and they can kind of swim a little bit on their own. They're not super strong swimmers, but they're mobile at some point. Now, sponges do not have germ layers. Germ layers are what develop first out when that zygote is formed. But basically, they do not have germ layers because they are organized at that cellular level, so they do not have tissue structure to them. All right, a little bit about this sponge body structure, and you are going to have to label the diagram that's on your set of notes as well. So the ostia are the first structure we're going to look at, and the ostia are the pores on the outside of the sponge where water goes in. Okay, so ostia in, they're going into the sponge. All right, so remember, once again, sponges are filter feeders. They bring in water, filter it out, and then push it back out another pore, and that pore that it comes out is actually called the osculum. So the osculum is the opening where water exits. So my tube sponges are the best example of this. Here's a lovely tube sponge. All right, so we've got this little structure right here. Notice on the outside, we've got all these little pores on the outside. That's the ostia. And then these big pores, these big openings in the top are the osculum. So that's where the water's going to exit. The water's going to come in through the ostia, out through the osculum, kind of like a little chimney pipe. We think about a tube sponge. Now, a normal sponge like this, what we think of when we think of sponges, ostia all on the outside, osculum are our bigger pores in which the water is going to exit. All right, choanocytes are another term that you are going to need to know, and this is what lines the inside of the sponge. So if we were to take a microscope and look at the internal structure of this sponge, we would see that there are tons and tons and tons of little things called collar cells. And collar cells basically just meaning they kind of line the inside of it. So these collar cells line the inside of that filtered chamber, and they trap food using a flagella. All right, so this little whip-like structure, that's what these little pictures right here. We've got a little collar cell, these little blue guys, and then they wiggle back and forth, and you're going to see this in the Jonathan Bird video as well. But this is what traps the food. They have sticky collars, and then that flagella moves all that water through it. All right, here's your sponge diagram. Get these labeled on that. You can pause this video to stop and label all of these different components. Notice there is one line. We're not labeling the epidermis on the sponge, but that is the outside layer. You have an extra line on yours. All right, a few more anatomical structures that we have already talked about. Spicules. So we did this when we did our symmetry stations, and we talked about the fact that sponges technically have an endoskeleton. So they technically have an internal structure that kind of gives them some shape and form. And that's in the form of these siliceous, so silica, or calcareous, that calcium carbonate support structures. And they're kind of shaped like little ninja stars or boomerangs. And these spicules are what line inside of that sponge. So we can see the spicule labeled here. Notice it's got like a little 3-point, kind of like a little ninja star, all inside of that sponge's epidermal layer. All right, amoebocytes are cells that actually move around inside of the sponge, and they secrete either silica or calcium carbonate in order to make the spicules. So amoebocytes are what make the spicules, and spicules are what gives the sponge a little bit of form, a little bit of shape, or else it would just kind of be floppy like a jellyfish. All right, so here's a picture of our spicules. We got some calcium carbonate ones on the bottom left. And then notice our silica ones on the top right are a little more see-through. Once again, if you want to see what silica looks like, go get you a pair of new shoes. Look at the little silica packet that comes in that, or sometimes comes in like new clothes. If you open it up, you do have to throw it away immediately. But you can see all of these little kind of glass-like looking balls inside of that silica packet, and that's the same thing that these spicules are made out of. All right, let's talk a little bit about how sponges reproduce. So how do we make more sponges? Remember that sponges are animals. So they've gone through that filter feeding process. They are sustaining life. They're going through cellular respiration, but they have to make more. So sponges actually go through both asexual and sexual reproduction. And through sexual reproduction, that sperm is produced in those collar cells. It's produced in those choanocytes. They can go through internal fertilization. So basically what they'll do is create a big giant sperm or egg volcano. It'll all come out through the osculum, the same place that the water exits. And then that sponge will release its gametes into the water column, and then those gametes are sucked in by a sponge through its ostia. So basically they are filter feeding and they are filter reproducing. They can also go through a process of asexual reproduction in the form of budding. They can actually grow an identical organism off the side. So case in point, we could have this sponge here grow on off a identical organism and an identical sponge off the side of that. And we're talking identical not just in structure and shape, but actually in genetics as well. Because remember, asexual reproduction produces genetically identical offspring. All right, so sexual reproduction, also some of them can be hermaphrodites, which is kind of cool. So they can actually have both male and female reproductive organs. They cannot fertilize themselves. So remember that sexual reproduction does take 2 mates. So they do have to find another mate in order to go through sexual reproduction. But it gives them the opportunity to kind of change to whatever gamete is needed within a population. So rather than having a giant population of all females and then no males around, because remember sponges can't walk, so they have to kind of sit where they, end up as an adult. This gives them the ability to reproduce within a population without having to go actually find a mate. So if they're hermaphroditic, figure out, what gametes are going to be released in order to go through that sexual reproduction. All right, this is the end of the Porifera notes. So make sure that you are getting that diagram completed as well.